Greetings dear viewers. In this video, I would like to explain about the Papillomaviridae family of viruses which causes warts and cervical cancer in human. I am Dr. M. Manigandan from the Department of Microbiology, Sacred Heart College, Tirupattur. Introduction. The Papovavirus family, that is Papillomaviridae, has been subdivided into two families. One is Papillomaviridae and the one is Polyomaviridae. Both these viruses are capable of causing lytic, chronic, and latent infections. The human papilloma viruses, which causes warts and several other genotypes of this, are associated with human cancer, majorly cervical carcinoma. The papilloma oma, which refers to the cancer causing ability of the viruses. Some of the important definitions in the papilloma virus infections are warts, condyloma, and papilloma. The warts, which are a small, hard, dry lump on the skin, which may be found on the face, fingers, neck, trunk, anywhere in our human body. Next one is a condyloma, which is a medical term used for genital warts. Genital warts are soft, non-cancerous growths that can be formed on the skin on the outside or inside of your vagina or anus or inside the cervical region. This can also be found on the lips, mouth, tongue, and throat. Papilloma, which means it is a benign tumor arising from an epithelial surface and usually known to grow on outward direction of our skin. Transmission. The virus has been transmitted from infected person to a healthy person through the direct contact with the warts. And the second mode is the sexual transmission. Nearly 100 serotypes of this human papilloma virus has been identified through its DNA homology, and these 100 serotypes may cause two types of HPV infection. One is cutaneous HPV and the other one is mucosal HPV. The high risk HPV and low risk HPV, which are found to cause a growth of a mucous membrane, that is over the skin, that is a low risk group, and the high risk group causes infections with the lesions or cancer. Structure. The papilloma viruses are found to be very much smaller in nature, which is non-enveloped with a icosahedral capsule. The capsid is of 50 to 55 nanometer in diameter, made of 72 capsomeres, which encloses the circular double-stranded DNA. And the DNA of HPV virus is approximately 8,000 base pairs, which encodes 7 to 8 early genes, E1 to E8, and it also encodes nearly 1 to 2 late genes, and the late gene L1 produces the viral attachment protein to bind with the host cell receptor. Replication. The HPV virus replication begins at the site of entry. The virus accesses the basal cell layers of the skin through breaks in the skin, any cut or abrasion or wound. The initial replication begins with the L1 protein, which is the viral attachment protein, and initiate replication by binding to integrins of the host cell receptors. The early genes of the virus stimulates the host cell growth, which also facilitates the replication of viral genome. The virus induced increase in the host cell number causes the basal and prickle cell layer, that is stratum spinosum, to become thickening, which leads to formation of wart, condyloma, or papilloma. As the basal cell differentiate, the specific nuclear factors expressed in different layers and type of skin, which promotes a transcription of different viral genome. Expression of this viral genome correlates with the expression of specific keratin. The late genes encoding structural proteins are expressed a terminally differentiated upper layer, when the virus assembles in the nucleus. As the infected skin cell matures and works in its way to the upper surface, the virus also matures and is shed with the dead cells of the upper layer. Pathogenesis. The papillomavirus infections may take around 2 years of incubation period. Initially, the virus infects and replicates in the squamous epithelium of our skin and causes warts. Secondarily, it causes infection in our mucous membranes, that is in the genital, oral, and conjunctival papillomas. The warts develop because of the virus stimulation of a cell growth and thickening of the basal and prickle layers of the stratum spinosum. The coelocytes are a characteristic structure of the papillomavirus infections, which are enlarged keratinocytes with clear halos around the shrunken nuclei. It usually takes 3 to 4 months for the wart to be developed, which is a localized infection, and it also can recur. The viruses causes benign outward growth of the cells into become a wart. The warts usually resolves and which takes due to the result of immune response. Dysplasia, which refers to the abnormal development of cells within the tissues or organ. It is a precancerous stage. Viral oncogenes. The high risk group of HPV can initiate the development of cervical carcinoma. The viral DNA is found in the benign and malignant tumors, especially mucosal papilloma. Almost all cervical carcinoma contain integrated HPV DNA with 70% from HPV serotype 16 or HPV serotype 18. The E1 or E2 genes promote integration of a viral genome, thereby it prevents viral replication, which does not prevent the expression of other HPV genes. The E5, E6 and E7 proteins of HPV 16 or HPV 18 have been identified as the oncogenes. The E5 protein enhances cell growth by stabilizing the epidermal growth factor receptor, which makes the cell more sensitive to growth signals, while the E6 and E7 proteins bind to the cell growth suppressor proteins, that is P53 and P105 proteins. The cell growth and inactivation of P53 makes the cell more susceptible to mutation, chromosomal aberration, or the action of a cofactor, and thereby it develops the cancer. Clinical symptoms. The HPV infections initially begins with the formation of wart, which is a benign self-limited proliferation of the skin that reduces with the time. The second one is the anogenital wart, otherwise called as condylomata acuminata, which occurs exclusively on the squamous epithelium of the external genitalia and perianal areas. Next is the genital cancer, which is a formation of a soft flesh colored warts on the genital regions, which are flat and raised. Sometimes these warts may be observed as a cauliflower shaped. These are identified with the coelocytic cells detected by Papa-Nicolau stained cervical smear, that is a pap smear. Other types of wart formation are the epidermodysplasia verruciformis, that is skin cancer. Next is the mouth papilloma around the oropharyngeal region, it causes cancer, and also in the laryngeal region it causes papilloma. Formation of tumors in the epithelial tissues of the larynx region. Different types of warts found in the human papilloma virus infections are the common wart, plantar wart, and flat wart. These are some of the symptomatic warts, formation of a cauliflower shaped, cristae shaped, mushroom shaped, papilla shaped, of papillary shaped, irregular shaped warts in the skin. Common wart, formation of rough raised bumps on the hand, fingers, elbow, and in between fingers. These common warts are painful, bleeding, and it is a unslightly causes irritation, which is small with tiny black dots on the center with clotted blood vessels. The second type of wart is the flat wart. It includes the formation of hard, smooth, flat topped, slightly raised lesions that can form in any part of the body. In children, it is usually found in the face, and in men, it is found in the beard area, and in women, it is found in the legs, which is observed as a flesh brownish yellow colored plantar warts. These are warts that develop on the plantar surfaces of the skin, that is on the sole or bottom of the feet. This is caused by the HPV virus invade through the tiny cuts or scrapes in the foot region. These are very much harder, grainy growth on the feet or heels, feet balls, and also found in foot region. This causes discomfort to walk, painful in nature, and this is spread commonly through the public swimming pools, communal showers, very much contagious in nature. Laboratory diagnosis. A wart can be confirmed microscopically on the basis of its characteristic histological appearances, which consist of the hyperplasia of the prickle cells and an excess production of keratin, that is hyperkeratosis. Papilloma virus infections can be detected in pap smears by the presence of coelocytic cells, that is vacuolated cytoplasm of the squamous epithelial cells. Its formation is rounded and occur in clumps. Molecular level identification by using ELISA PCR helps us to differentiate between the various serotypes of the virus. And papilloma viruses do not grow in ordinary cell cultures. Treatment. The warts reduces spontaneously upon time, which may take from several months to years. Usage of salicylic acid over the warts reduces skin irritation, and the innate stimulators such as imiquimod, which is a cream used to reduce the redness and the swelling. The other drugs including cidofovir selectively kills the HPV infected cells by apoptosis, and the usage of podophylox reduces the burning or itching sensation on the genital region. Usage of trichloroacetic acid reduces the irritation of warts over the palm region. Physical removal of the warts by laser surgery and other treatment methods to reduce or remove the warts by loop electrosurgical excision procedure by using heat, freezing with liquid nitrogen, that is usage of extreme cold, cervical colonization, which is a removal of cone shaped warts from cervical region. Preventive measures. Immunization with either a tetravalent vaccine made of HPV type 16, 11, 6, and 18, or a divalent vaccine HPV type 16 and 18. This vaccine is recommended majorly for girl children starting at 11 years of age. Other methods of prevention by picking of Avoid picking of warts with nail. Avoid biting of the nails. Proper precautions during sexual intercourse. Wearing of shoes or sandal in public modules. Avoid public showers and swimming pools helps us to prevent us from HPV type infection. Thank you for your patient listening. If you like the video, kindly share and subscribe to our channel. Thank you.